Topic is about energetics of ATP synthesis. In previous videos, we have seen that exactly how that electron transport chain is occurring, but when this electron transport chain, the free energy is released and that must be conserved in a form that ATP synthase can perform energy coupling. So when this electron is transported in this electron transport chain, ATP should be generated. So this energy of electron transport is used to drive proton out of the matrix by the complex 1, 3 and 4. So we have already seen that when electron is transported through complex 1 and 3, 4 proton are pumped out from matrix part of mitochondria to the intermembrane space. Same way complex 4, but here only 2 proton are pumped out. They are work as a proton pump, that is complex 1, 3 and 4. The proton gradients thus generated is maintained across the inner mitochondrial membrane till electron are transferred to oxygen to form water molecule. So the electrochemical potential of this gradient is used to synthesize ATP molecule. So basically that matrix part to that intermembrane space, electron is transferred out, and that is the main function of this electron transport chain. So according to the estimated free energy of synthesis, it was presumed that around 3 protons are required per ATP synthesized. Hence, when one NADH transfer its electron to oxygen, so NADH starting point of this complex 1, then pass to the coenzyme Q, then complex 3, there is a 4 proton again pump out, complex 4 again 2 proton is pump out, so 4 plus 4 plus 2, total 10 proton are pumped out. Now this would account for the synthesis of approximately 3 ATP. Similarly, the oxidation of 1 FADH2 is accompanied by the pumping of 6 proton, accounting for the 2 ATP. Simple that FADH2 from the complex 2, no proton is pumped out, complex 3, 4 proton is pumped out, and again complex 4. So 4 plus 2, total 6 proton out, and that can be accounted for the 2 ATP. But recently proved that the actual energy production is less because there is always leakage of protons. When NADH is oxidized, 10 hydrogen ions are pumped out, that is corrected. But according to recent finding, where there is a leakage of protons, one NADH may generate only 2.5 ATP and one FADH2 may generate only 1.5 ATP. Previously we have thought for the NADH 3 and for FADH2 2, but as per the recent finding, NADH will generate only 2.5 and FADH2 will generate only 1.5 ATP. So one molecule of glucose will generate only 32 ATP, which is quite lower as compared to the previous calculation. So that also we will look. So energy efficiency of glucose oxidation giving 32 molecule of ATP and palmitate, that is giving 106 molecule of ATP, is given as a 34% and 33% respectively to the overall energy production. So what are the rest of energy? So out of total energy production of glucose, only 34% that is trapped as a ATP, that is total 32 ATP. Same way palmitate generating 106 ATP, that is only 33% of total energy production. So what about the rest of the energy? Rest of the energy will be utilized for the heat production inside the cell. So traditional value and new value. So NADH will generate 2.5 as per new, FADH2 will generate 1.5, glucose which is considered previously 38 ATP but at present only 32. Same way acetyl CoA, one molecule of acetyl CoA completely oxidized in a TCA cycle which is generating 3 NADH, 1 FADH2 and 1 ATP. So substrate level phosphorylation ATP formation is not changed, but what are the energy calculation as per that NADH FADH2, you have to remember NADH 2.5, FADH2 1.5 ATP generation. So one molecule of acetyl CoA will generate 10 ATP molecule. Same way palmitate, that will generate total 106 ATP molecule. So apart from that energetics, you have to remember about PO ratio. P means phosphate, O means oxygen. So the PO ratio is defined as the number of inorganic phosphate molecule incorporated into ATP. Inorganic phosphate incorporate into ATP for every atom of oxygen consumed. So when a pair of electron from NADH reduces an atom of oxygen, 2.5 mole of ATP are formed per 0.5 mole of oxygen consumed. So for 2.5 mole of ATP formation, 0.5 mole of oxygen is consumed. So in other words, the PO ratio of NADH oxidation is 2.5. So 2.5, that is the PO ratio for NADH. Same way PO value for FADH2, that is 1.5. So that is PO ratio. That value you have to remember, sometime ask as a MCQ. Now here site of ATP synthesis, which is in a ETC. So what are the site of proton pumped out? So this 1, 3 and 4, that is considered proton pump, and that is considered site of ATP synthesis. So site of not actual ATP synthesis, it is site of proton pump out. So this proton is pumped out in a complex 1, that 4 proton pump out, complex 2 no proton is pump out, complex 3 4 again, and complex 4 lastly 2 proton is pumped out. So that about the energetics of ATP synthesis, PO ratio and site of ATP synthesis. Now what are these H plus is in accumulated in this intermembrane space, further that will be utilized for the ATP synthesis. In detail we will look in the next video.